single file, right? You have how many chromosomes here?
4.
4 chromosomes. And then once it separates, the daughter cells, how many chromosomes will it have?
2.
2 and 2, 4. So that will be here 2 red. And here on this side, 2 red and 2 blue. Right? Because this is a chromatid separate. Once the chromatid goes here, that becomes 1 chromosome. Once the chromatid comes here, that is 1 chromosome. Same thing for this one and this one. Right? So how many chromosomes at the end? Right here?
4.
4. How many chromosomes here?
4.
4. So notice mitosis, you begin with 4, you end up with 4.
And each
Each cell.
has 2 homologous chromosomes.
Here, yes, you will have, but they are not replicated. Yes.
Okay.
One from mom, one from dad.
Okay.
Okay. In this case, if it is a human cell, right? Somatic cell, mitosis. So you have 4 chromosomes. Here they have been replicated, sister chromatids. Those are basically telling you that it has been replicated. Here they are non-replicated. Okay. But you have 4 chromosomes, 4 chromosomes, 4 chromosomes. Mitosis end result, 2 daughter cells that are equational, same number of chromosomes. Okay. Diploid, diploid. Okay. Same number, daughter cells, identical. Okay. In meiosis, here you have 1 cell, and now here you have these homologous chromosomes, 1 from mom, 1 from dad. Okay. Well, now they are next to each other, double file. Okay, 2 lines. These are the tetrads right here. Okay, sister chromatids, non-sister chromatids. Genetic rearrangement, genetic exchange, charismata, right? Boom, 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 right? And then it divides. And here, how many cells will it have? I mean, how many chromosomes will it have? This cell? 2, right? It takes these 2 red ones over here. So you will have 2.
What did it be like?
What's that?
What about
It could, yeah. So let's say that this one is changed right there, right? Will it mix right there? That means genetic rearrangement, okay? So what we have right there. Okay? And how many, so you begin with 4, and how many chromosomes on this cell? 2. So you begin with 4, you end up with 2 daughter cells that are reductional, haploid, 4, 2, 2, okay? Again, this one has the genetic rearrangement. Meiosis 1. In meiosis 2, you begin with how many chromosomes?
2.
2 chromosomes, right? Let's take this cell right here. What happens in both cells? Now they separate. Now in this case, the sister chromatids separate. So now you have this. Okay? How many chromosomes here? 2. How many chromosomes here?
2.
So it is equational. Begin with 2, end up with 2. Same as mitosis, right? And here, the thing that is different, it is that they are non-identical. Here, they're identical. Here in this case, they are not. One will have genetic rearrangement, the other one will not. Or what will have, but they are different. Okay? Depends, let's say this one was here, and so. Okay? Right, any questions?
that was for how sperm cells are made, right?
Meiosis, yes. Sperm and egg cells. Yes. Those are going to be haploid. Yes.
You said meiosis 2, there's 2 chromosomes, 3 chromosomes, and 3 chromosomes. So it says, ahora I'm gonna start doing four chromosomes.
Right here?
Yeah.
No, these are two chromosomes, one chromosome, one chromosome. This one is sister chromatids. Like that. These are sister chromosomes. Remember, here the homologous chromosomes separate.
Yeah.
It remains the sister chromatids together. So it's still one chromosome. Once the sister chromatids separate, that's when it becomes the chromosome. Because it's going to go to another cell. If they are joined together in the same cell, it is one chromosome. Yes?
What about the step where you do the 4 identical daughter cells? Sorry, 4 non-identical daughter cells.
That will be this one, right? So this one is for this. If you do this one as well, then you have another two that are different.
Okay.
So there are 4 at the end. So this is for this. For meiosis 2. Meiosis 2 will be the same thing, right? Then it will have 2 daughter cells. And at the end, you will have meiosis, these 4 daughter cells. Okay. Non-identical, different from one another, and different from the parent cell. Okay? That is the point. So division of meiosis 2, pretty much the same, right? Prophase 2, singular products form. Then you have here in late prophase 2, the chromosomes is still composed of the 2 sister chromatids, right? Joined together with sister chromatids. That is, for example, here, as mitosis, what this one is not identical, right? Here, mitosis, they are identical. Meiosis 2, they are not. And they move toward the metaphase plate that we have right here. So this is meiosis 2. Pretty similar as mitosis, right? But, again, they are not the same. They are not identical. You can see here already the genetic exchange that has taken place. Okay. They are looking pretty nice like Spider-Man, like that. Okay. Have you seen the Spider-Man movie? No. Yeah, so it's pretty. I need to go and see it. Yeah. Well, anyways, yeah. But they separate, right? Anaphase 2. They separate. Sister chromatids separate in anaphase 2. Okay. And then you have telophase 2 and cytokinesis. So at the end you have these haploid daughter cells, which are non-identical, different from one another, and different from the parent cell. Metaphase 2. So these sister chromatids, they are arranged at the metaphase plate, and because this crossing over that has taken place in meiosis 1, prophase 1 to be exact, the 2 sister chromatids of the chromosome are no longer identical. I keep saying this time and time again, right? So make sure you know this one. That one is going to be exact. These sister chromatids in anaphase 2, metaphase 2, they are not identical. The kinetic course of the sister chromatids, they are attached to the mitochondrial extending from opposite poles, so this will be like similar as what you guys have done here for mitosis. And then on phase 2, the sister chromatids will be separated. So now here they move as the newly individual chromosomes toward the opposite poles to be divided and to create the resultant daughter cells. In phase 2, again, the chromosomes are divided at the opposite poles, then begin to condense, nuclear form, so cytokinesis, division of cytoplasm. And then you have at the end of mitosis, the 4 daughter cells with haploid number and unreplicated chromosomes. Okay? And each daughter cell is genetically identical, distinct. Not identical, okay? Distinct. Not the same. From one another or from the parent cell. So, key distinction. Mitosis conserves the number of chromosomes, which is equational, and it produces 2 daughter cells that are the same, identical. Okay? Meiosis, it is reductional, it produces a number of chromosomes even from the blood, from 2 sets, to being haploid in 1 set of chromosomes, and they are different. Okay? So here just basically just a simple diagram that shows you the difference between mitosis and meiosis. So here you have the prophase, the condensed metaphase in mitosis lined up in one file, as I mentioned before, and then you have the separation of the sister chromatids, and now you have the creation of the daughter cells. Equational. Then you have here in meiosis, you have here the chiasmata, that is the X shape right there that happens in prophase 1, the chromosomes pair up, the synapses, creating the genetic rearrangement, genetic change. And then you have the metaphase 1, the homologous chromosomes, they pair up with one another, next to each other, double file, 2 lines, as opposed to mitosis. Then here, the homologous chromosomes separate. Okay? The whole chromosome with the sister chromatids separate. Now you have here the daughter cells. Now they are reduction. And then you have here meiosis 2, where those sister chromatids, they will separate. Okay? And then at the end, you have 4 daughter cells which are haploid and non-identical. Here, another summary, right? This basically tells you more about it and where this takes place and all that. DNA replication, only once in both. The number of divisions, 1 and 2. The synapses only occurs in meiosis, meiosis 1, prophase 1. Number of daughter cells, here you have 2 and identical, here you have 4 and non-identical. And the role in the animal body, here for mitosis you have the growth and development, tissue renewal and repair. And for meiosis you have the production of the gametes, sperm and the eggs. If you know this, you are good. Okay, you know this table. Right. Key events that I mentioned that takes place on meiosis. I will ask you like something like this on the short answer questions. Okay, either write or identify. Like more like the mechanisms between, or like this, the distinction between mitosis and meiosis. So make sure you know that all of them, all these 3 of them, they occur in meiosis 1. Okay. Prophase 1, the synapses, the crossing over, the chiasmatic, right? That's where the genetic variant takes place, right? That's number 1. Okay. So if I ask you this like this in the short answer questions, what are the 3 mechanisms that are distinct in mitosis and meiosis? Draw them or whatever. That would be your 2 points. Okay. 1, the chiasmata synapses, genetic rearrangement, right? Synapses, chiasmata, that happens in prophase 1. 2, the metaphase, the homologous chromosomes, they pair up next to one another. So not single file, but double file, 2 lines, right? Draw them, right? Not just 1 line of chromosomes, but 2 lines pairing up next to each other. That's what you guys did right here. The tetrads, right? Sister chromatids and the non-sister chromatids. And then number 3, the anaphase 1, how the homologous chromosomes, they separate instead of the sister chromatids in meiosis 1. Okay? Draw those things, identify them, or describe them. Okay, well, that will be that.
When we're drawing them, like we will like how we color it, like make the difference between like the alleles joining together, like the blue and the red joining together. Prophase 1.
Yeah, something like this. One prophase 1, genetic rearrangement. Metaphase 1, the homologous chromosome pair up there, and the anaphase 1, separation of the homologous chromosomes. So draw something like this or better. I would say better. This is just like for example. But once you draw something like that, then try to be detail oriented and also like more description. Yes, as you mentioned, how the genetic rearrangement takes place. All that. Yes. So those 3 points would be that, right? Or you would have to tell me those 3 points. Right? Draw them. Alright, so here you have the sister chromatids. They were added, that hold together by the cohesion, right? So that's what allows them for these single chromosomes to stay together to the meiosis 1. How they're separated, that's because they're still together to sister chromatids. These are protein complex called cohesins that are responsible for this union, for this cohesion. In mitosis, these cohesins, they're cleaved at the end of the metaphase, separates chromosomes and cut them. In meiosis, these cohesins, they are cleaved along the chromosome arms in anaphase 1, that is separation of the homologous. And at the end of the centromeres in anaphase 2, that's where this is the chromatism is separate. So it comes in parts, how they are cleaved. Alright, so enough about mechanisms and about distinctions of meiosis and mitosis. So now let's see about variation. Okay, this genetic variation that you see in the human population. Mutations, those are changes in the organism's DNA, and actually that are the original source of genetic diversity. Whenever you have these shocking alleles, that's what you have there, right? The sperm and the egg cells combining together, the alleles of the mom, the alleles from the dad, joining together, and so forth, right? Anything? Yeah. But mutations, those are the ones that add like new alleles. So new alleles added to the So these mutations create different versions of these genes, these alternate versions of the genes that are called alleles. So I mentioned that before. And I will mention them again in the next sessions. And this reshuffling of alleles during the sexual reproduction produces this genetic variation. Now, this behavior of chromosomes during meiosis and fertilization, that's what is responsible for most of this variation that arises in this generation. Like I mentioned this, how they can combine these independent assortment, how they are lined up next to one another. And these are the 3 mechanisms that contribute to this genetic variation. The independent assortment of the chromosomes, that's what I mentioned right there, I will mention in a minute. The crossing over, the exchange of those genetic material in the prophase 1, and the random fertilization. Think about that, for example, right? Imagine that from that pool of sperms, which is something about like 200 to 500 million sperms, right? Once you know your dad and your mom, they got really happy, right? That number of sperms right there,
Whoa.
the only one, random fertilization, was you. You are here, right? One of them was the winner, it was you, right? One of them, one sperm out of like 200 to 500 million sperms, that is you. The lucky one, the winner, right?
No, I thought it was the second one.
Which one second?
Like, isn't it that the first one doesn't fully make it, and the second one's the one that makes it?
Yeah, no, but it will be that, right? Random fertilization, right? It will be that sperm right there. But it will be the lucky one that basically fertilizes sperm. That lucky one, right? The main sperm that it does that. Random fertilization will be one out of 200 to 500 million sperms. Random fertilization. Then you have all this independent assortment. Then you have this crossing over. So for these homologous pairs of chromosomes, they will arrange randomly at the metaphase 1 of the meiosis. Okay, that is known as independent assortment. So each pair of these chromosomes, they will sort the chromosomes from your mom and from your dad, right, on either side. And these homologous chromosomes, they will then form these daughter cells. Questions over there? Which one? Plan B?
Yeah, we're talking about birth control, like how does that work? How does it kill so many sperms?
Like, so,
I mean, I'm just curious, like, if you're saying it's like thousands of sperms,
Mhm.
how does one thing, like Plan B, birth control, like, how does that kill all those sperms?
yeah, okay. Well, I mean, I think we can touch that more on the next session, and we will talk more about that, like, yeah, on the next module. Yeah, the next module, right? So just put your thumb down, yeah. Okay, so this is the assortment, right? So you have these possibilities for these chromosomes. They are to arrange in this manner or in this manner. Let's say you have 4 chromosomes, right? And these 2 are from your mom, and these are for your dad, the same thing over here. So they could either arrange here, on the left side, all the ones that are from your dad, and the right side, all the ones that are from your mom. Or it could be mixed, like here one from dad, here one from dad, right? Either one, okay? So the arrangement, it doesn't matter, because if you have them in this way, then by metaphase 2, and then by daughter cells metaphase at the end of the anaphase 2, then we'll have these possible combinations of the daughter cells, as opposed as if they were over here in this manner, mixed, okay? Then the combination will be here. Okay. So keep on right here. Yes, question.
So, like, it's just a random question. Well, I think you really said, so the chromosomes already, like, when the sperm is landing on the egg, it's already having its, like, own DNA?
Yes.
So the chromosomes or the DNA inside that sperm is already, like, set on the gender? Or?
The sperm is what is going to determine the gender, the sex of
Oh.
the new baby in the
Oh.
organism. Okay.
Okay.
And yes, it will have its own set, unique DNA, as opposed to the parent, to the dad, or to the mom. It will be unique. Yes.
Oh.
It will not be the same as yours. It will not be the same as the dad, but it will have its own DNA.
So nothing will change, like, in the process of,
In the,
like, nothing will change in the process of, like,
in the process.
yeah, like, you know, like, yeah.
She was asking if copulation changes the gender of the offspring. So like, no, nothing would change. 'Cause nothing would change. The thing that matters is whatever DNA is inside the sperm or the egg. So even if you have 2 sperms going, those 2 sperms have 2 different DNA, like 2 different end product. So doesn't matter what happens when you copulate. The only thing that matters is that the sperm and the egg meet each other and becomes
I ask because if DNA determines like a lot, like people can get extra chromosomes or like
Oh,
less
yes,
chromosomes.
yes. We're going to see that.
Oh,
Yeah,
okay.
yeah, yeah. But if I understood this correctly, like you are saying if the gender can be changed by different process, different things?
Mhm.
Well, yeah, that's a subject for the next sessions where we could see that different factors could alter, but it will not change, right? It will not change in the sense that your genetic makeup, it is what you inherit, right, from these cells, right? There could be like certain factors such as hormones, right, or different conditions of disease that can affect the expression of it, right? But the genetic makeup, what you inherit, is what you inherit. That's what you have. The sperm and the egg. Once they combine, they have a unique set of genome, DNA, and that's what you have. That will be expressed. So it's not like, oh yeah, yeah, my wife is pregnant. Then put this like metal thing around it so it will work. No, it will not. It will not. Not the case.
Wait, so it can what?
And so you can, make the baby so the baby will be boy, right?
Oh, it's not.
They're like some
But don't they have something now, like where you can choose your gender? Like, isn't there like people who can't really have their kids? Don't they take out the sperm and like choose?
Well, I mean, you are now going to more like specific techniques such as like CRISPR-9 as well to modify certain genes or to choose like in vitro fertilization. But in there, you have to do some screening, right? So it is not, it's not like that you are changing it, right? For example, here once the egg and the sperm that are fertilized, each of those two cells they have the genome, right? Once they are mixed, that's what you have. That's what you have. That's what it will be expressed. All right, so that is the number of independent assortments, right? Independent assortment, remember, that's how those. We will talk about those key points, for the next sessions. But for this one, the independent assortment, that's pretty much like the number of chromosomes that it has and how they will be separated. Okay. For example, there, which chromosomes will be inherited one cell, which chromosomes will inherit the other cells. Well, all these cells from the chromosomes from the mom will be inherited in one cell, and all the chromosomes from the dad will be in another cell, or they will be mixed, right? Those are like independent assortment. So here you have how to calculate that. For humans, the n number is 23, right? That's the haploid number. For the possible combination for the independent assortment, that's basically this 2 to the n, where n is the haploid number. So for humans, will be 2 to the power of 23. So that would be around more than 8 million possible combinations just from those number of chromosomes that you have, and how you will create these gametes that will have to be fertilized once they join with one another. This independent assortment. So just by independent assortment, you have like more than 8 million possibilities of you becoming you. Added to that, the random fertilization, like one sperm in like 200 to 500 million, right? The independent assortment for just different possible combinations, you can have like 8 million possible combinations. And now you have as well the crossing over, right? The recombinant chromosomes, the genetic rearrangement that takes place in the prophase one, right? So adding or shuffling those different alleles that you have from the mom and from your dad. So this chromosome crossing over these homologous portions of the two non-sister chromatids, they will take place, right? And this also contributes to genetic variation that you see in this DNA, in this DNA of this new organism that it will be from the parents to the single chromosome. So that's what you have there, this crossing over. Here you have the non-sister chromatids that are held together by the synapses, and then here you have the chiasmata, the X-shaped centromeres right there, and now they are separated. And then you will have the daughter cells, which are non-identical. Okay? Non-identical to one another, and non-identical to the parent cells. So you have the random fertilization, you have the independent assortment, and you have the genetic rearrangement right here. So at the end, that's like adding like possible combinations, like around like 70 trillion. So all of these crossing over, that's more operation. Each cycle has unique genetic identity. So yeah, like you are like 1 like 70 trillion, right? So Ladies, you know, whenever the guy comes and tells you, like, you're 1 in a million, so no, you're not right. Get your math right. I'm 1 in like 70 billion. 1 out of 70 billion. Guys, if your lady comes and tells you that you're 1 in a million, just say yes, honey. Don't get into trouble. Do not correct her. Happy wife, happy life. As Will Smith, not. Anyway, yes. Right, just to cover this, random selection results in accumulation of these genetic variations affected by the environment. So those traits are affected by the environment. Those are the ones that are going to keep passing on. And sexual reproduction contributes to this genetic variation or genetic mutation, right? So key points from this. Right, I know it was a long slides, many good questions that we're going to cover them in like the next sessions and the next module. But for this one, make sure you know the difference between mitosis and meiosis. Those tables, those diagrams, make sure you know that they're going to be there, okay? And make sure you know the 3 key points for the differences in mitosis and meiosis, the one that I mentioned, prophase 1, metaphase 1, anaphase 1. Right? Make sure you know that, right? And make sure you know this, right? The genetic variation, those key points that add to the genetic variation, okay? Chromosomal, crossover, fertilization. Okay. All right, any questions? No? Okay. All right, so let's pass the exams back, and after that,